Hello, everyone. Thanks for being here. We'll just wait a few more moments as people connect. New people coming in. All right, then. Just give it a few more moments. It seems like a few more people are joining us. Thanks so much for being here. All right, it looks like it's about that time. So good afternoon, everyone. Again, thank you for being here with us today. A few small pieces of administrative stuff before we get started. The chat is disabled for this session, so please put all questions, and we hope you have many, in the Q&A box, and we will look forward to a lively discussion at the very end. So we are at the half hour, which means it is my very great pleasure to introduce Professor Michael Webster. Dr. Webster is a vision scientist at the University of Nevada, Reno. He received his PhD in biological psychology from UC Berkeley in 1988 and was a postdoctoral fellow at Cambridge University for 6 years before joining the UNR faculty in 1994. At UNR, he is a foundation professor and director of UNR's COBRE for Integrative Neuroscience, which is currently in their phase 3. So we know that's quite an accomplishment around here. His research is focused on the mechanisms and functions of adaptation in the human visual system, and his work has been supported continuously by NIH since 1994. So today, he's going to tell us about adaptation and visual experience and the ways in which our sensory processes are constantly recalibrating into the characteristics of our environment and thus affecting our perceptual judgments and profoundly impacting our experiences. So please join me in welcoming Dr. Michael Webster.
Okay, well, thank you so much, Hillary. And I want to thank you all for joining me today and giving me this opportunity to speak. It's an incredible honor for me to be able to connect. And I'm really excited to be able to connect and interact with other Cobras. As Hillary mentioned, I'm at the University of Nevada, Reno, and even though that's where I'm based currently, I'm on my first sabbatical in 25 years, and I'm actually talking to you right now from a hotel in Jodhpur. And it's about midnight here, so I'll apologize beforehand if I sound a little groggy. But anyway, I've been doing some really fun work over here for the semester. But back in Reno, when I'll be back in January, I do direct our COBRA for integrative neuroscience. And when I try to tell people who don't know about COBRAs what's really going on with these centers, I actually like to point out that, in fact, Nevada's first COBRA was in 1906, long before even the NIH, and it was based over here in the northeast corner of the state. Turns out that cobre is Spanish for copper, and this was a center for research excellence in hauling copper ore. And so they built this railroad there, and they set up a town to make this happen. And even though the topic is very different from what a lot of us are working on in our Cobras, the strategies were actually really similar. One of them, as you know, is basically to take some really promising young recruits and then pair them with some old geezers to try to help them learn to dig a little deeper. And then the other aspect of the centers is really to build the infrastructure that you need in terms of core facilities. And so for our first COBRA, that included a general store and a hotel and actually 2 different saloons. And everything started off really great. It was a really booming town and center for a while. I think they got up to a population of about 150 people. But then somehow things went wrong and there was a lot of discord and infighting. And if you go to COBRA today, even though it was a bustling, productive community at the time, it's now really fallen into neglect. And in fact, all that's left there is a few abandoned core facilities and some tracks that just dead end in the sagebrush. So anyway, this is sort of the image that keeps me up at night stressing, because this is not, I'm sure I speak for all COBRA PIs in saying this is not what we want our COBRAs to look like after the grant funding ends. And so we all spend a lot of time wondering, what are we going to do to make sure that what we're trying to create is actually sustained? In terms of the current Cobra that I direct, we're in our phase 3. And I think a lot of you can sympathize when I say, thank God there's not a phase 4. But anyway, we're in our 14th year, so we're really getting to that point where we're focusing really hard on what are we going to do to make this a permanent, lasting impact on our university and state. My COBRA is very broad. It's basically integrative neuroscience, so I don't have a specific disease model, for example, or focus. It was really to build research capacity in neuroscience at my university. We've been able to support over 24 junior faculty, either as projects or pilot projects. Most of them are still here, and most of them have gone on to independent funding. We've also established a bunch of core facilities. These include a neuroimaging center, which brought the first fMRI facility to the state for research. A cellular and molecular imaging facility that has, for example, 2 photon and confocal microscopes. And I was told it was a mistake, but I think it was a good idea. I actually proposed a new core as part of phase 3 just to address the explosion in data science and artificial intelligence. And so we have a neurocomputing core that we started as part of that. In terms of trying to make a lasting impact, we've tried to build out from the COBRA. We now have undergraduate and doctoral programs in integrative neuroscience. We've created an institute for neuroscience, which is sort of a continuation of our administrative core, just to be sort of the hub and support center for all the neuroscience initiatives on campus. That institute has already brought us 2 training grants, a T34 for undergraduate, and then I think the first T32 from NIH training grant in the Mountain West IDEA states. That was all work of the institute director, Jim Kenyon. And finally, we just got a really large supplement of over $1 million from NIH that's going to the NBRE and our COBRA to actually merge our computational cores. So we have to figure out how to leverage that in the very brief time that they give you to do those. So anyway, that's my center. Again, in terms of my research, as Hillary mentioned, I'm a vision scientist, which basically just means I want to understand how we take the information coming in from our eyes and process that in order to understand the world around us. And my own particular interest is sort of things like how does that information get adjusted to the environment we're in. And I've been doing this at UNR since 1994. And this is actually the state sign that greeted me when I first arrived for my faculty job there. And as someone studying vision, you can't imagine how thrilled I was that we have this long history of vision in our state, and we're actually proud enough to put it on our welcoming sign. At the same time, I was really surprised by that because I'm pretty sure that when I entered Nevada, I was the only vision scientist in the entire Great Basin. And so I wondered what happened to all the other vision researchers. And I got kind of worried when I got up close and saw all the bullet holes. But anyway, this sign really symbolizes to me how I think about the perceptual process as this prospector is looking out on this beautiful panorama and wondering how he's going to pillage it. At that same time, this landscape, these visual world in front of him, I believe is strongly shaping the way that his senses are working and affecting what he's actually seeing. And those are the questions I'm interested in, is how is your vision really a product of the environment and the world that you're actually living in? And the main way that I look at this, there's many different forms of sensory plasticity, but I mainly focus on relatively short-term examples I'll show you in a matter of seconds. I can change the way you see the world just by exposing you to a different stimulus. And we know that, like all the senses, vision is highly adaptable. So for example, if you look at the center blue cross in this figure here, try to keep your eyes centered there. Then when I switch to a blank slide, you might, especially if you blink your eyes, you might see an after image, even though the demo says it only works if you have faith. But anyway, if I switch here and you kind of blink your eyes, hopefully you can see a face there. So it's kind of easy to understand what's going on there. If I showed you the image on the left, then the part of your eye that was seeing the dark part of the screen thought it was dark outside. This is a very local adaptation. So it has to get more sensitive, just like a camera has to become more sensitive in dark light. The part of your eye that was seeing the bright white screen thinks it's bright outside, it has to become less sensitive. And so if I switch now to a blank screen, then the part of your eye that's more sensitive is going to be responding more. And so it'll look bright there and conversely dark on the other side. And that is basically, so what you're seeing is the negative of the image that you adapted to. And this is very common of what we call visual after effects. These after effects basically look less like what you've adapted to because you've become less sensitive to what you've adapted to. Finally, it probably didn't look as crisp as this middle image here. It probably looked a little more blurry, and that's because even though I told you to fixate that point there, your eye's constantly jittering around, and that's going to smear the image out on your retina. So it's going to look a bit more blurred. So anyway, that's kind of all there is to it. And once you know how these are made, it's kind of fun to make your own. And since somebody already tried it with Christ, I decided I'd try it with the devil. So this is a picture of my daughter. And if you, again, look at this cross here, then each part of your eye is independently adapting to the local brightness that it's seeing. And again, if I flip to the blank screen and you blink your eyes, you might see an image of her. Okay, the next point I want to emphasize is that this adaptation, even though it's common to talk about simple things like adapting to the average light level, it's actually happening throughout the brain. I personally believe that everything you see, you're actually also adapting to, and it's happening in very similar ways. And so this means that basically this adaptation is really a general principle that's affecting everything you see. And I think it goes beyond perception to how everything you kind of even think about or experience in your life. But anyway, we can show very similar forms of adaptation just by using different kinds of stimuli. And as an example, when I showed you that light adaptation to those previous images, we know for a lot of reasons that that's happening right down in the eyeball itself and actually in the photoreceptors that are absorbing the light. So it's a very early sensitivity change. But there's other kinds of changes that we know are happening later in the brain. Now, this image is probably not going to work on this video, but it's supposed to be an expanding spiral. And if it does look expanding to you, then you're basically adapting to that motion. And when I flip to a static screen or image on the next slide, it should actually look like it's contracting. And that's because very rapidly, again, your brain has adapted to a particular property of the world, this movement. And so you become less sensitive to that particular property. And we know for a lot of reasons that this adaptation is actually happening not in the eyeball, but at the back of your brain where your primary visual cortex is, and probably even in later areas that are specialized for motion perception. So the point of all this, again, is I can basically just use different stimuli to adapt different stages of the brain and then try to figure out what's going on at those different stages. And because of this, this adaptation has actually been called the psychologist's electrode, because instead of actually sticking an electrode in and recording from single cells, we could again show you stimuli, look at how your perception changes, and from those changes, try to work out, okay, what was in that part of the brain that got adapted and is changing, like how sensitive was it for different properties of the world, how many of them are there, and so on. So it's been a really powerful tool in perceptual psychology for basically understanding how the senses work. And just as one example of this, which I'll get back to a bit later, this was a study I did long ago when I was a postdoc, where we basically just had people adapt by looking at a colored light field, basically, that was flickering between two colors like red and green or blue and yellow and so on. And then we just measured how sensitive you were to all of these different colors. And what we found is that no matter which direction you adapted to in this color space here, you lost sensitivity to that. So if you've adapted to red and green, you become less sensitive to red, green than to blue, yellow, and so on. And so this suggested that at the level where this adaptation is happening, and again, we think this is happening up in the cortex, not in the eye, you could show that basically you had channels for color mechanisms in your brain sensitive to color that were tuned to many different color directions. And this turned out to be very different from kind of the classical theories that we had of color, which are that you only have two cells or types of cells sensitive to red, green, or blue, yellow. So anyway, that's how adaptation has largely been used as more as a tool for trying to understand how vision is built. But the questions I've mainly been interested in are what are the states of adaptation that you're currently in? So I've shown you again that in a matter of seconds, I can change your perception. What kind of changes are happening as you're walking around in your world right now? What's different about your vision and when you're in a room versus going outside or traveling to a new place? How are you recalibrating for that? And so those are the kind of real world problems I've been interested in. And the main point that I want to make here is that this adaptation is always basically adjusting you to match the properties of the world outside you. And I think this has really profound implications for things like whether you and I see the world similarly or differently. And the basic idea is that even if you and I are very different, say, in terms of our brains, our physiology, our optics, we're going to see some aspects of the world in very similar ways as long as we're adapted to the same world. We're both going to be calibrated to a common kind of experience. Conversely, if you and I are exposed to different worlds, that same adaptation is going to cause us to have divergent percepts and start to see some aspects of the world differently. So I want to go through several different examples where we've actually tried to look at these. And the first cases are basically all in color vision. I mainly think of myself as a color scientist. And if we want to think about color, it's useful to use something like a perceptual color space, which tells you about what are the different dimensions that are important of color. But these spaces are kind of complicated. And so instead of using the classic or conventional uniform color spaces, I made up my own. I actually did this for my um, postdoc supervisor in Cambridge for his Festschrift a year ago. But anyway, I made my own uniform color space, and what's different about mine is it's actually based on uniforms. And anyway, like most color spaces, this one is a circle. And at the center of that circle is basically the neutral or the gray point. And so this is kind of the center again of your color experience. And then one way other colors can differ is things like in their hue. So they could, for example, vary between red and green or blue or yellow, or you could even have these intermediate colors. So all of this we call basically the hue of the color, whether it's red, green, or blue. But colors can also vary in things like saturation, how strong that hue difference is, how far away it is from gray. And it can also vary in lightness, like how bright or dark it is. And the point about these, again, this is sort of a perceptual representation of how the brain codes color. And the point is that in this entirely colorful family of characters, there's only one that actually matters, and that's that gray point there, that neutral stimulus there, because that's the anchor of the entire system. And in fact, all that really matters in this family here is how you're related to this person, right? And how many steps removed you are and how, what direction it is. But basically, it's all relative to this one point. So the question is, where does that gray come from? What actually sets gray in your head? And we think that basically it's done by just, again, adapting you so that the what looks gray to you is literally just the average color of the world that you've been exposed to, or more specifically, the average spectrum of light. And so color, again, is extremely adaptable. If you look at these four squares here, if you just look in the center, then again, each part of your eye is adapting to a different color in the four boxes. And when I flip, you hopefully can see some color after images. So again, in just a matter of seconds, you're adjusting what looks gray to you on this screen here. Excuse me. One way we've tried to look at this in a real world context is say, well, different people have different average colors reaching their eye just because, for example, the lens itself is colored. And it turns out that as you age, your lens is getting yellower and yellower over time. And that's just because light is actually bad for your eyes, and it's basically building up this pigment inside the lens. If you're lucky enough to live into your 90s and not have cataract surgery, you're going to have a very dense brown lens. And what that means is that the light reaching your eye is going to be very filtered. There's going to be very little short wavelength light getting back to your receptors. And here I've actually simulated this. If this was the image as seen by a young adult, then if we add the lens of an older adult, basically, again, you're losing all those short wavelengths. The world should look really yellowish to you if you're not adjusting to that. But in fact, if you ask older observers, what does the world look like to them? What color looks gray or red and so on? They show remarkable agreement with younger individuals, even though they have a very different visual system. And again, that's because they're being calibrated to the same world, which is basically discounting or removing the filtering effects that are unique to them. One extreme case where we've looked at this is in cataract surgery. So this is a case where you have a very dense lens. You take that out, flip in a clear interocular lens, and now again, you're seeing the same world through a very different visual system. And so what we did was basically track how do their color percepts change after surgery. This is actually done with my colleague at UC Davis, Jack Werner, and his student, Peter Delahunt, and they didn't even let me help with the surgeries. But the point of the study was basically, again, before surgery, you have very little sensitivity to these short wavelength blue lights. You put in the interocular lens, you're suddenly flooding their eye with blue light, and they say the world suddenly looks a lot brighter and a lot bluer. But we could then track the changes in their color percepts over time. And surprisingly, what we found here was this very long-term drift. And so basically, this is showing that what looks white to them, what they say looks gray, is slowly drifting over time back to what it was before the surgery. So again, they're over time, they're starting to see the world the same way as they did before the surgery, even though they themselves have changed because, again, you're being calibrated to the world outside you. Another case where you can see this in your own visual system is the fact that you have a second filter in your eye that's called macular pigment, and that's right over the center of your vision, so right where you're directly looking. It also filters out short wavelength blue light. So right where you're looking and fixating, you're seeing much less short wavelength light than you are in the surround. And here again, I've just simulated this, that if this is what colors look like to you right where you're looking, then this is how blue they should look off to the side because you're no longer looking through that filter. But once again, the world seems to be pretty stable. If you look at a white wall, you don't normally notice a spot. It turns out you can occasionally see this. I don't know how well this is working on your screen, but this is a great illusion that someone just sent me where if you look at any one of these spots, hopefully it'll look purple and all the other spots look blue. And that's happening again because when you're looking directly at it, it's being screened by this pigment. So you're blocking out more of the blue light and the red is leaking through more. But normally, again, you don't notice those kind of effects at all, even though they're really sitting there right in front of you. And in fact, we've shown objectively that what looks white to you in the right where you're looking and off to the side remains very similar, despite these enormous differences that are happening within your eye at these different locations. We've even shown that if you do these color after effects, these adaptation demos, even those are calibrated to the same white point. So again, if I show you a red, then a gray looks green or vice versa. The point that doesn't look show an after effect, the white is basically the same in both places. So this is really a physiological null point or neutral point that's been calibrated within your eye. The last example I want to give for color is looking at color deficiencies and extending this to say, well, to what extent do people who even have a color deficiency, are they experiencing the world the way that we would predict from their change in their visual system, or are they actually more similar to us than you would have otherwise imagined? To understand color deficiencies, my favorite example is the TV license in Britain. When I became a postdoc there, I was really surprised to find out that I actually had to buy a license to watch TV. And they were really expensive at the time. And I remember I was at a point where I was asking myself, do I want to watch TV or do I want to get my kids a Christmas present? And I ended up putting a license in their stocking. But they still do this. I'm not making this up, but It turns out that if you want to watch a color television currently in Great Britain, it costs you 169 pounds to do that. So that's around $200 a year to watch that. But if you want to watch a black and white television, it's only 57 pounds. I don't even know where you'd get one of those anymore. And I don't know how they come up with these numbers, but the amazing thing is if you look at the ratio of these two numbers, it's almost exactly 3-to-1. So they're charging you 3 times as much to watch a color television as a black and white TV. And this is really fantastic. This is a policy that's actually based on science, something we're not even doing anymore. And the idea behind it is, well, this makes sense because, in fact, we know the way we see color is because we have 3 different kinds of receptors that are sensitive to different wavelengths of light. And you're basically getting 3 times as much information. So it makes sense that you should charge someone 3 times as much if they're getting that color. The part of this license that doesn't make sense, and again, I'm not making this up, this is still what's their policy in the UK, is that if you're legally blind, you get a 50% discount. And what that means is that in England currently today, it costs more for someone without vision to listen to a color television than it does for a sighted person to watch grayscale TV. And that seems so bizarre, it makes Brexit seem like a good idea. But anyway, it turns out vision is not all or none. There's all kinds of varieties of color sensitivity. There are some people who only have 2 kinds of receptors, so they're only getting 2/3 as much information as most people. There's other people called anomalous trichromats who still have 3 kinds of receptors, but 2 of them have sensitivities that are very close to each other. And so their ability to distinguish, say, reds from greens is actually weaker. And you can go on the internet and you can ask, well, it's common to find websites where they say, well, what do the colorblind see? And what you'll typically see is an image like this middle one here. And what they're doing here is they're taking an original image and then they're just filtering it by the shifted pigments or the loss of the pigment in these color deficient observers. So basically, again, you're losing that red-green sensitivity. But what this image doesn't show is that these individuals are also adapting to their world. They've actually had a lifetime to do it because this is a congenital condition. And even if the receptors are giving a weaker red-green signal, there's no reason why post-receptoral cells shouldn't be expanding that difference and recovering some of those red-green differences. And in fact, when people have started looking at this, they found that their claims of their color experience, even though they can't see the differences on the screening test, their actual experience of color is not nearly as impoverished. So here's a study where they just asked people to compare red versus green differences to blue versus yellow differences. Based on their color losses, you'd expect reds and greens would look really washed out, but they report they're almost normal. And the problem with this kind of experiment is it's really telling, but you can't be sure. Are they really seeing it similarly, or are they just have they learned that everybody else is labeling colors a certain way, so they start to do that too? And so we've been trying to look at objective evidence for this. And one thing we've used is fMRI, which is a way of non-invasively measuring brain activity. And basically just showing them colored patterns and asking how strong are the neural responses in these early visual areas to these color patterns. And what we found, in fact, is in the primary visual cortex, the first cortical area, there is a real loss of color sensitivity. The responses are really weak, but by the time we get to the next cortical stages in the visual system, they're almost indistinguishable from normals. So this is consistent with, again, a real amplification that's basically causing this individual with a very different visual system to potentially be representing the world in their brains in similar ways, just because their brains are being calibrated to the same world. Now, the last example I want to give on this is basically looking at spatial vision or form perception. And here again, we know that everyone's very different. None of us has perfect optics. There's always some kind of imperfections, which means the image on the eye is always going to be blurred to different extents. If you're an optometrist, you think that, well, a purely focused image is the best you're ever going to do, and then things are just going to get blurrier. But from the perspective of the brain, you could be off either way. Things could be represented as too blurry, or they could actually be overrepresented in terms of the fine detail. And so I think myself that sort of the stimulus that looks focused to you is the equivalent of gray in color space. It's the neutral point that's calibrating despite your optical imperfections, letting you see the world itself as physically focused. And so we started studying these effects. And here's an example where you can adapt to this property of the world. So if you fixate again, the center spot here, then these two images are both physically focused. Now keep looking at the center spot. The image on the left has been blurred. The image on the right has been sharpened. And when I switch back to the original images, the one on the right should look briefly more blurred to you. And so again, in a matter of seconds, just what looks focused is actually changing. And what we found, in fact, is that if you're looking at a blurry image, it actually starts to look better focused to you. And what did look focused now looks too sharp. When we first started doing this, we were just filtering the images in ways that were kind of arbitrary. But we can now basically look at to what extent are you actually adapted to the specific optical properties of your eye. So if you have a particular kind of refractive error, are you specifically adapted to that? And one way we can do this is use a system called adaptive optics, which was borrowed from astronomy. And basically, you can measure the optical aberrations, the blur function in somebody's eye, and then actually use a deformable mirror to bend the waves of light so that they basically correct for that. And with this, you can show perfectly clear image in someone's eye, or you could introduce the blur of someone else's eye. And so this is work that I did with Susanna Marcos, who at the time was at the Institute of Optics in Madrid. It's not something I could do at all, but she's remarkable at it. And again, what we could do is basically take the images through different people's eyes. So these are shown how blurry these different individuals' images are based on the optical quality of their eye. This person's got really good vision. This person has poor vision. And with this adaptive optics, we could literally remove whatever your blur was and then introduce somebody else's blur and ask, what does the world look like through this person's lens? And one thing we found is that the image that looks correctly focused to you is almost perfectly predicted from the amount of blur that you're normally used to seeing. And we can also show that this is not just that that's because you're used to it. This is literally how your vision's calibrated. And the way we show that is to take somebody's blur function, which is shown by this colored patch here, which is just showing, again, the errors in the eyes optics. We can either exaggerate that and make it worse, more blurry, or we can actually turn it down, make it sharper than this person's ever seen, and then adapt to those images. And what we found, in fact, is again that the adaptation is really if you adapt to an image that's sharper than you've ever seen, then what you're used to calling focus will look too blurred or vice versa. And again, this is evidence that what looks focused to you really is a calibration of your brain for the different spatial structure of images. Okay, now those again were all examples where we know that people are optically or neurally different from each other, yet their perceptions aren't as different as you'd predict from that. And again, what's going on here is that this adaptation is in a way removing your own idiosyncratic properties so that even though we have these differences, we're starting to perceive the world in similar ways because the job of perception is not to tell you about your own properties. It's to use whatever visual system you have to get your best guess of the world outside you. And what it says again is that even if people have very different visual systems, they might actually have very similar experiences of the visual world. But the converse of that is that if you were to take the same person and put them in different environments, then that predicts that they should start seeing things differently because now their brain is adapting to a different set of stimuli and it should, again, start calibrating to different set of neutral points. And so I now want to go through some examples of those. And related to this, when the Barbie movie came out, I guess a couple of years ago, I actually got contacted by Scientific American, and they wanted to ask me what would it be like to live in a pink world like Barbie. And it turned out this is something that I think about all the time. And so the question again is, would Barbie actually see differently from you because she's in a pink world that's different from your world? And if you saw the movie, you might also ask the question, well, does it matter what Ken sees at all? But anyway, we normally don't really, even though we know that there's individual differences, it's hard to kind of imagine that someone can be looking at something and see it differently. And it's only when an image comes out like this that people are really struck by the idea that two people could be looking at the same thing and have very different experiences. So I hope most of you remember this image of the dress here that came out about 10 years ago, where about half the people thought that these were white stripes and golden stripes, and the other half said that they were blue and black. And this caused real viral discussion throughout the world, and especially among the vision science community about what was going on with this. And the explanation that people quickly came up with is it's not that people were seeing the colors differently, it's actually they were attributing them differently. So the idea is that if you saw the dress as blue and black, that might be because you think you're looking at the photograph in direct sunlight. So you think that the blue is actually coming from the dress itself. If you instead you think you're looking at the image in shadow and these two images here are exactly the same, then you say that you think that, okay, the blue is actually coming from the lighting, not from the fabric. So the fabric instead looks white to you. And this is a problem we have to deal with all the time. In fact, in vision, that the world is full of, for example, of things like shadows, where in this side lit face here, you almost hardly notice that half of it's in shadow. You're basically filter that out. Whereas if I make these mirror images here, the differences become really apparent. And the point is, if you're actually looking at discounting these shadows, well, shadows are also typically blue. And that's because usually you're blocking the yellow sunlight and you're just getting the light coming in from the sky. And so we know that people are very good at sort of ignoring the blueness of shadows. But if you flip that around and make it yellowish, then it no longer looks like a shadow. It looks like there's actually some material here. Here's another example we looked at where basically the room on the left is, I can easily myself see that as a white room in blue light, but if I flip the color here, I can't see that as a white room in orange light. This now looks to me like an orange room in white light. And similarly here with these coins, if I just increase the color, then these on the right here, the top still look like silver coins to me in blue light. But if I flip to yellow, they actually look like golden coins in white light. And anyway, when the dress came out, we were actually the first one to have a publication of this because we've been studying these effects all along. And as soon as the dress appeared, we tried it with that. And even when I made the colors of the dress 3 times stronger, 3 times bluer, I was still getting about a third of the people were saying that this is a white stripe. Again, not because they don't see the blue, but because they attribute that blueness to the lighting. The minute we flipped it over, almost everybody agreed that the color with the dress was yellow and there was no ambiguity with the lighting. So anyway, that's just an example of individual differences, and it's kind of a digression from what I mainly wanted to talk about, but I did want to mention this study because I'm really proud of it because it's the only work I've ever done that's been important enough to get written up in 17.com. Anyway, getting back to adaptation, we've known from other studies that if you stick a graduate student in a red or green room for hours at a time, sure enough, their color vision will change. And in our studies, what we wanted to do was ask, well, does the real world actually differ enough in color so that depending on where you live, you might see color differently just because the color of the world you're in is actually different. And these are studies we started a few decades ago, looking in the Western Ghats of India, conveniently close to where my wife grew up. And so when I first went there, these are the hills outside of Mumbai, and some of the most green, lush part of the world I'd ever been. And this is the same valley just a few months later. There's monsoons here, and the minute the rain stops, the vegetation dries up and it becomes a very different color world. And moreover, we were able to work with people who are mainly farmers. So they're literally out immersed in these color environments. And we were interested in, well, what are their color percepts? And could those even change, for example, with the seasons? So anyway, to do these studies, what we did was pack up some equipment, and then we hired some help. And I've been coming to India now for about 40 years, and it's been some of the most fun I've ever had and some of the most amazing people I've gotten to meet. Like these are Indian sadhus or holy men. This was at the Kumbh Mela, which is a major Hindu festival. And through my wife, I could talk with them. And it turns out that they're sitting around talking about the same kind of things that I talk about in conferences, like what's the nature of our senses? Are they just providing us with an illusion, or do we really sense reality and things? And in fact, the only real difference, I think, between me and these Indian holy men is that they're not locked into the cycle of rebirth and suffering that people like me are in, because they don't have to apply for grants. But anyway, in these studies, what we've done is basically to take calibrated cameras and basically measure what are the actual spectra in different parts of the world or different times of the world. There's again work that I did with my wife, and so I always want to show this slide to just acknowledge my collaborator and the love of my life. You really can't buy a better spectroradiometer. Um, and then once we had those measurements, basically we could take them back into the lab, and then we could expose people to sets of colors that were drawn from these different outdoor environments. So this would have been an arid scene, this would have been a more lush scene, and so on. I already showed you, again, we knew that from other work that if you adapt, say, to a blue-yellow environment, you're going to become less sensitive to blue-yellow compared to other colors. And so we could show that in a very short time, if we just adapted people to this natural color distribution, that they would selectively adjust their color perception in ways that were very predictable. And we also wanted to know, well, can we see evidence that people in these environments are actually seeing color differently? This is the first time I've done field work like this. And, we wanted to know what this person thought about the color, but it was really hard to do that without somebody else looking on. And sometimes there was a whole village looking on. And if the guy chose the wrong color, then the crowd could get pretty hostile. But anyway, these are the kind of measurements that we got from this. And what's being plotted here, basically the colors people would point to in their own languages when we ask them to identify things like red or orange or yellow and green and so on. And the first point is you can see, like with yellow, that all these different groups in India and in Reno at the bottom here are very similar in what they describe as yellow. But within that category, there's very clear shifts. So the average Indian observer would say that the average observer in Nevada, their yellow is too greenish, and we would say that their yellow is too orangish on average. And these differences, again, could represent differences in, again, the colors that they're actually calibrated for. I couldn't actually see a seasonal variation in color. That's one of my goals on my present sabbatical, is go back and try to do it the right way. But it turns out a couple years ago, there was actually a study where they showed this in York, England. And they showed that, in fact, there's differences in what people say as pure yellow between summer and winter. And that was a big surprise to me, because I lived in England for 6 years and I didn't know that they had seasons. Okay, the last example I want to give on individual differences and how those are driven by adaptation is in face perception. When I started doing this work, there was an actual portrait of the Queen that was being painted, and it was by the artist Lucian Freud. And she sat for this painting 80 different times, and this is what he ended up with. Lucian Freud, by the way, was Sigmund Freud's grandson and was considered at the time England's greatest portrait artist. He passed away, unfortunately, a few years ago. But anyway, this is what he came up with. I never heard what the Queen thought, but but the British were not very happy about it. And these are some of the reactions from the tabloids. And the one that I really liked was this bottom one that she should have known what to expect when she hired him, because this is his self-portrait. And the point is that he has a very particular style. So when we see these paintings, we're kind of overwhelmed with the style. But imagine him painting it, looking at this day after day. He probably sees this face in a way that you and I never would. So we started looking at whether you actually adapt to faces the way you do other things. And to do this, we took a normal face and then we could distort it different ways, like pulling the eyes together or pulling them apart. And then do this by distorting them either horizontally and vertically and make all these faces. And when I did this, I told my ex-friends at Cambridge that I actually discovered something amazing. No matter how you distort a normal face, it starts to look more British. But anyway, what we found is that you adapt to faces just like you do everything else. So here's a demo of this. This is the original face. If you look at this contracted face, then the longer you look at it, the less contracted it looks. You can almost feel your mind pulling it back out. And if this is becoming a normal looking face to you, then the original face should look too expanded. The eyes should look too far apart. And in fact, that's what we found. This is actually an example of what we call a high level effect, because it's happening at a very high level in the visual pathway. And one example of this is a study where they repeated our experiment, but they adapted to a face tilted this way and then tested this way. And they show that the after effect actually rotates with the face. So that means that the sensitivity changes are occurring at a stage in your visual system where you're representing the world kind of independent from your own viewpoint. And we know that, in fact, there's a large network specifically involved in face processing, which we now think is adaptable in very similar ways. The next question is, well, do real faces differ enough so that you would actually get different states of adaptation depending on who you're surrounded by? And so we started studying these with lots of real variations in faces, and everything we tried turns out to be very strongly adaptable, whether you're looking at, say, male versus female, young versus old, and so on. Here's another example of how striking these effects can be. So just keep looking at this fixation cross here. And the faces in your periphery are actual photographs. They're not being doctored at all, but they might start looking like really grotesque caricatures. I didn't discover this effect. It really upsets me because I was always looking right in the center. But it turns out these effects are really whopping off to the side of you. And what's going on here again is that each face, as you start adapting to them, as each face comes up, what's getting exaggerated is just how does that face differ from the previous ones you've been seeing. So you're seeing that kind of after effect really exaggerated. And again, we've seen these effects for almost every face, every aspect of faces that we judge about people. And again, to me, what's really interesting about this is this face adaptation seems to be calibrated. What you perceive as faces seems to be calibrated in very similar ways as color vision. So we think, in fact, that the way we represent faces is not on an absolute scale, but we're always judging how does this face differ from this internal prototype or average that I have, which is like our gray point in face space. So once again, it's a relative judgment. And the point is that one example of this, for example, is that average faces tend to look more attractive. And this has been shown just by taking a bunch of individual faces. If you blend them all together, then the average is typically rated as better looking than any of the individuals that went into it. And the issue again is, well, all of us are exposed to a different average set of faces, right? Depending on where you live, who you interact with, and so on. So each of us are probably walking around with a different prototype that we're using to judge faces with. And this can explain many, I think, aspects of how people respond to things like faces. So I'm, again, a vision scientist. I actually get stressed if I get too many synapses into the brain. But I think this explains a lot of things that have been talked about in very different cases like anthropology or social psychology about how do your judgments of attractiveness change. Here's a famous quote from Malinowski, who said that when he moved to the Trobriand Islands, at first he didn't agree with what they thought were the most attractive people, but he said over time he started to agree with them. And that's always been attributed to saying that his attitudes about attractiveness were changing. But I think what was going on is that his criterion was staying constant, average is attractive, but what was average to him was actually changing in this new environment. Anyway, this could even explain why people are attracted to people that look like themselves. We might all be adapted to our own familiar characteristics. And it could even explain why we're attracted to dogs that look like us. Okay, I think I'm getting a little late on time, so I'm going to rush through this last bit. But if I need to stop at any point, please pull the plug. Um, the last point I want to make is that I and a lot of other people in my field have tried really hard to find natural worlds where we can see what are the natural states that we're in and how is vision calibrated to the natural environment. But a big problem is very few of us live in natural worlds anymore. It's completely artificial constructions that we live in. And so this raises a whole bunch of really interesting questions about what kind of adaptation states are we in now that we have all this new technology and can basically create the worlds around us. One case where we're starting to look at this is in radiology. So these are radiologists spend hours a day staring at these very special images that have their own visual characteristics. And they're making really important visual judgments about these. So we've been asking, do radiologists actually adapt to these images and can it affect the kind of diagnoses they make? And we've been looking at mammograms, and basically one of the first things a radiologist does is decide, is the tissue that I'm looking at either dense, meaning it's hard to see through it and harder to diagnose, or is it more fatty? And so what we did was basically ask, are those judgments even affected by adaptation? And that's important because if the radiologist thinks it's a dense image, they're more likely to call for follow-up tests and so on. And here's an example. This is a blend between a dense and a fatty image. So they're the same on the left and the right. Now I'm adapting you to a dense image on the left, a fatty image on the right. Keep looking at the dot, please. And when I flip back, the image on the right should look more cloudy or dense to you. So we've shown again, that's a very adaptable property, just like all the other properties that we've looked at. We've recently shown that even trained radiologists show very similar adaptation effects. And so these biases, what it means is that, in fact, how you're judging the current image, the current patient might be affected by the visual properties of the screening that you were looking at previously. We've also shown that this can actually help in detecting abnormalities in the images, because one function of adaptation is the idea that it's basically building an expectation or prediction about the world and filtering that out, leaving all of your brain resources to basically pick up what are the unusual, suspicious, surprising properties of the world. And that's exactly what a radiologist is doing. Anyway, we've shown that if you first adapt to the images where you're trying to look for an abnormality, it actually helps you. Last point is, another nice thing about radiology is you can actually go back and look retrospectively at diagnoses that were actually made and see what kind of behaviors happen as people are looking longer and longer in this batch of images. And the most interesting case was a study in England where basically they have two readers look at every batch of images. And what they were trying to do is control for fatigue. They thought after an hour, you're going to be getting worse because you're getting tired. And so someone's always looking at sort of those endpoints, either at the beginning or the end. Instead of getting worse, what they found was people were actually getting better. So this is inconsistent with fatigue, but it is consistent with the idea that as you adapt to these images, you become better, more facile about seeing them and making judgments. I'm going to skip that for time. Now, the very last point that I want to make is we actually understand enough about these adaptation processes and things like color that I can actually predict what the world would look like to you if you did move to a different place and adapt differently. And so I have a model that's basically based on how the visual system adapts to color, and I can build adaptation into that model. And these are my predictions of what these environments would look like to someone who's completely adapted to them. So in an arid environment, the greens really do stand out more because they're more novel properties of the world. And as a more extreme case, this is my prediction of what Mars is going to look like when your grandchildren move there. So we call it the red planet, but it's not going to look red to them because, again, what's gray or red or green is really just the average spectrum you've been exposed to. If you're living in a reddish world, it should look gray over time. And this is not just a hypothetical question, right? Soon we're going to actually be able to test this. As you know, lots of people are trying to figure out how can we send people to Mars. There was actually this Mars One mission a few years ago. They're going to send 12 people to Mars on a one-way trip to establish a colony there. Over 200,000 people applied to get on this one-way flight. And it turns out that one of the finalists is actually from Reno, Nevada. I don't know how they picked these people. Maybe they thought, if we send someone to Mars from Reno, at least they won't get homesick. But the question I want to ask is not about why they picked her, but why did 200,000 people want to leave Earth on this treacherous journey? And to answer that, I want to go back a little closer to home and talk about the Donner Party. And as I think you must know, because I noticed they spent a lot of time walking through Nebraska, that this is an immigrant group that basically started off in Missouri and trying to get to California. And through a series of bad decisions, they ended up in very close to Reno, Nevada. So I feel a lot in common with them because they basically got as far as Reno and then realized they probably weren't going to get out alive. But anyway, right after they left, the mother of one of the Donners actually died, and they buried her in Kansas. And then her grave was discovered by James Kleiman, who was this famous mountain man. And you can picture him showing up at this grave in beaver skins. And what does he do? He pins this incredibly philosophical passage on what on earth would have led this person to leave the comforts of civilization. And he said basically that the human mind is just built to seek novelty, and that's the whole goal, and that's what's really the driving force in life. And my point is that novelty seeking is really built right into even the fabric of how your senses work. And so if I can end on this last final slide, if you just fixate this cross here, then if you fixate very carefully, those purple dots might fade away, and all you'll see is that green spot. That green spot is the afterimage when one of those purple spots disappears. But otherwise, those purple spots might completely disappear to you. You don't even notice them because your brain already knows about them in a way. You've adapted to them. And all that's really filtering up to your awareness is the sudden change or the unexpected properties of the world. So I literally believe that everything you can see around you, everything you notice is in fact a visual after effect. And I know that it probably sounds like it has very little to do with substance abuse. I really apologize for that. But I think that this is a principle that's not just about the senses, but really about how the mind works in general. And it's really important for understanding to what extent two people have sort of shared experiences. Sorry, there's kind of a rat running around this hotel room. Um, but anyway, I'm done. And I just want to thank everyone again for giving me the opportunity to speak. I'd love to form connections with your Cobra. And as you're getting closer to phase 3, I'd love to help in any way I can without having been through that process. And I apologize for running a bit late. Thank you.
Wonderful. Well, thank you so much for being here. I think I will focus on some of the questions. Folks, if you want to stick around, I'm happy to stick around a little while. Is that okay with you, Mike?
Yeah, that's fine.
Okay. So one of the questions that was asked in the Q&A box this is actually about the first part of your talk, which was about just having a successful Cobra, and any advice for faculty who are wanting to build a center or institute at their universities.
Yeah, my advice, I guess, is it's a lot of work and it's not going to work out the way you want, but it is. The nice thing about a Cobra is they're big enough that you really can make a difference. And and so it's a real opportunity to really make a lasting and dramatic effect. And again, we've been able to branch ours off in many different ways. Even though the Cobras aren't about teaching, we've been able to start training programs and things. And so I feel really good about that. I remember going to a Cobra meeting, and there was a senior Cobra PI to me, and his advice was to keep the main thing the main thing. And, or the main thing is to keep the main thing the main thing. I don't think he came up with that, but that's very good advice that it's easy to get kind of lost in the details. And it's really important to just keep your eye on that big picture that you really are doing something important for your institution and your state. This is a really great program. I'm really glad it's continuing to be funded because I think it's really making a difference for a lot of people.
Awesome. I will also,
But if
oh,
you are putting together a Cobra, I'd be very happy to give more specific advice in terms of what worked for us and maybe more importantly, what didn't.
very good. I'll also remind folks if you'd like to put additional questions in the Q&A box. I have some back channel questions though that were sent to me in another mechanism, so I'll ask a couple of those if you don't mind. One of them, folks are wondering, because our center is a substance use center, do you know of any substances that may affect these adaptation processes in meaningful ways?
Um, yeah, that's a fantastic question. I don't actually know how it interacts. So the adaptation itself seems to be a really robust, like it seems to be largely unaffected by aging, even though there's so many other changes that happen with aging. And I myself think that's because it is really fundamental that you can't be born calibrated correctly. And again, I think this is on so many levels that I think it's like a psychological principle. It's not just a sensory principle. Um, I don't know enough about the biophysical mechanisms to know and about how that might be affected by substance abuse.
So this is hopefully going to turn into a question and not so much a comment, but this, this is not my area of research, but this talk has really gotten me wondering a few things related to this individual difference piece about adaptation. And so, like, I've had 10 years now to realize that I'm never going to see that dress as anything but white and gold. And during one of your examples, when you had the contracting and expanding annulus, and you said, I don't know if you guys can see it, and I could see it super well. But I also like get severe simulator sickness. I think my night vision is kind of questionable. Facial recognition is iffy. Do you think that there are like implications of this in the clinic or for various disorders? Or it seems like you have ways to, I'm going to say, treat people who are, maybe struggling with adaptation. Do you have thoughts on that?
Yeah, so I think so one thing you can do, and people are starting to look at, is whether you can teach people to adapt better. So one example is in virtual reality, one of the big drawbacks is motion sickness. And a lot of people don't like it because of that. And people are starting to develop regimens where you basically can adapt over time for that. I've had funding from places like Johnson and Johnson who make a lot of contact lenses and want to know, are there things we can do to help people adapt to a new kind of prescription or like a different kind of contact lens and things. So people are definitely thinking in terms of how you could like harness adaptation to help and maybe speed up those processes. Um, I don't know of cases the other way. To me, it's a really fascinating question of whether there are large individual differences in adaptation and whether those differences are sort of a global, is there just some underlying common mechanism that's not working, or is it really specific to particular um, types of adaptation or plasticity? But I'll say a general problem, I think, in perception and cognition is we all talk about plasticity. Again, adaptation is just one form of it, but there's all kinds of other like developmental and um, recovery from damage and things like that, and we and learning and evolution or all forms of plasticity. And we don't, as a science, I think, have a good unified theory of what plasticity really is, how many different mechanisms there are, how they differ. Like, is it in terms of the mechanism or just the principle? But they've almost just been defined operationally. When we see a change, we call that plasticity. So to me, that's a real important gap in the field. And until we address that, I think it's really hard to then map that onto understanding, say, what's going on in these different populations and which forms of plasticity we should be targeting.
Very cool. All right, well, I think that it's 1:30 in the morning for you, is that right? And about 5 after for us as well.
And it's 1:05. India is actually a
One,
half hour, which is
okay,
strange, but I discovered it's because they span two time zones, so they just use the average of the two for the whole country.
split the difference. I like it. Well, thank you again once again for being here. Oh, we've got getting accolades in the Q&A also, if you want to check those out. But good luck with that rat, and we hope to hear a lot more from you.
I have to admit, it's caught my attention, but yeah,
Well,
well,
thank you
thank you
very much
all.
for your time.
I'd
And
love to
thanks everybody
follow
for being here.
when anyone has questions, COBRA related or science related. I'd love to talk with you all. And thank you again.
Thank you so much. We really appreciate it, Mike. Thank you, Hillary, for moderating. Thank you, everyone, for joining us. This was such a fascinating conversation. I look forward to more conversations and seeing ways that we can collaborate in the future. So I hope you have a really great evening. Thank you, everyone who joined us. I hope you have a great afternoon, and we'll see you soon.
Sounds great. Thanks,
Thank
everybody.
you, everyone. Thank you, Mike.
Bye.
Bye.